Hi, my name is Trung Ky and I currently go to Alhambra High School as a senior. I work under the guidance of Ni Zhang at the USC School of Pharmacy, and I made this presentation on October 12, 2009 for my Biomedical class. The title of my project is Identifying the Contributor of Beta Cell Proliferation with Different Markers in Mutated P10 Knocked Out Mice or Cre-LoxP mice, in which I will explain later. Now, I've been motivated to do this project because my sister has type 2 diabetes. Secondly, 23.6 million people, or 7.8% of the US population, have diabetes. 17.9 million are diagnosed, while 5.7 million people aren't. Now, another reason for doing this project is to alleviate the economy. As you can see, the US spends on average of $174 billion every year to treat patients. Our lab is to try to find a cost-efficient treatment to diabetes. And because there's no cure, we need more research on what causes the insulin in the pancreas to produce. So far, there are only treatments to treat patients, but no cure. Now, as you might know, there are two types of diabetes, type 1 diabetes and type 2 diabetes. Type 1 diabetes is insulin-dependent diabetes. Now, insulin gradually decreases over time due to the attack of the immune system. Secondary diabetes occurs due to cystic fibrosis, surgery, etc. Type 2 diabetes is non-insulin dependent diabetes, when insulin is not utilized efficiently or correctly. Now, you hear insulin, but what does insulin really do? It regulates the fatty tissue's intake of glucose. Otherwise, if we have too much glucose, glucose will flow into the bloodstream. Now, too much glucose in the bloodstream can cause dehydration due to excessive urination, weight loss, which results in loss of calories, and hyperosmolar non-ketotic diabetic coma. And that's when a person is unable to drink water to make up the fluid that is lost. Also, it causes body damage or nerves and small vessels in eyes, kidneys, heart, and hardening of the arteries, and that is called arteriosclerosis. And all of this can result into pain. Now, people with diabetes are those that seem overweight, obese, or people who inherit it from family members, or people with high cholesterol, or elderlies, or even smokers. Beta cells can be found in the islets of the pancreas. Now, if there's more beta cells, there's a higher possibility of producing insulin. P10, which stands for phosphatase-intenstin homologue, regulates the process of cell division. It is considered a tumor suppressor, and it could be found between the Cre-LoxP recombination. Cre stands for cyclization recombinant. It knocks out the P10 from the DNA recombinant. It causes the linkage of the LoxP segments, in which I will show you later. Cre-Lox knocks the stop codon and links the ROSA and LAC-Z segments together. What is the importance of LAC-Z? It identifies the cells that the beta cells make. BRDU, or bromodioxyuridine, helps detect the proliferating cells in cell tissues, and it identifies what the beta cells are making. You may have learned PCR. PCR is polymerase chain reaction. It creates a template of the DNA strand, goes through genotyping, and it detects the specific and or wanted enzymes within any organism. Samples taken are normally used in electrophoresis. Now, here's an illustration of what I was talking about. Cre is initiated by the rat insulin promoter, or RIP. Remember that Cre, or cyclization recombinant, knocks out the P10 LoxP and links the LoxP segments together. In the ROSA LAC-Z segment, it knocks out the stop codon to link the two segments together. In our lab, does P10 regulate beta cell proliferation? And what kind of cells do the proliferating beta cells come from? We hypothesized if P10 is depleted from the LoxP gene, the beta cells will proliferate, and that these proliferating cells come from the cells that use to produce insulin. Now, here's an overall of what we're going to do. First, we are going to knock out the P10 from the Cre-LoxP combination to obtain the wanted alleles for the experiment. Next, we will knock out the stop codon from the ROSA LAC-Z series to obtain the recorder of the beta cells. Finally, we'll perform a procedure of the BRDU label with different markers to detect those proliferating cells. So here's our first procedure. We breeded 2 mice with the given alleles and given traits, and we got the trait and alleles of the daughter in red. After 1 month, we genotype the mice. To do this, we extract the DNA by digesting the tail with proteinase K, which is like a dissolver. We run through PCR with Cre, Laxi, and P10 enzymes, and then we run it through the electrophoresis gel. When the mice are 3 months old, we give them 1 milligram per milliliter, or cc, of distilled or deionized water for 5 days. We then kill the mice and fix the pancreas onto slides. We use immunohistochemistry, or IHC, to stain 2 types of antibodies. We clean the bacteria on the slides with xylene and ETOH, and this is called deparaffinization. We add 30 microliters of antigen retrieval solution for 23 minutes to expose the tissue. We added 30 microliters of BRDU primary antibody for 15 minutes, wash in PBS, and add 30 microliters of power block for 10 minutes. This power block blocks proteins for the next antibody to be stained. We use the MOM blocking kit, and there are further information on or instructions on that. We label BRDU with a specific marker. We stain the antibody marker with the green and BRDU with red. We add 1 drop of avidin blocking PBS, then biotin. We repeat these steps to stain again. Keep in mind that IHC can only do 2 stainings of antibody. As you can see, BRDU is labeled in green, insulin is labeled in red, and GAPI, which isn't what we're concerned with, is labeled in blue. On the top, you can see that there's P10 mutant and wild type. P10 mutant is the mice with the P10 knocked out. Remember that P10 is a regulator of cells, so P10 mutant is the mice without P10. The wild type has P10 in it. Remember that BRDU is to detect where the beta cells are at. In the P10 mutant, we can see that there are lots of beta cells when P10 is knocked out, while in the wild type, there are very few. In other words, when P10 is knocked out from a DNA recombinant, more beta cells proliferate. When P10 is present, then there are fewer beta cells. Remember that BRDU is in green. The red, however, are different markers that we tried marking the islets of the pancreas with. The point of the marker is to see where the proliferating cells are coming from. Glucagon, somatostatin, PP, cytokeratin, and amylase were the enzymes that we used to mark these areas in the islets. If we see that BRDU and glucagon are overlapping, it indicates that the proliferating cells are coming from that marker. However, in any of these pictures and results, we see that none are overlapping. From the first illustration, we see that P10 does regulate beta cell proliferation. However, in the second illustration, to answer what kind of cells do the proliferating beta cells come from, it's still undetermined and inconclusive. Of course, every experiment is not perfect. We had several different errors. During genotyping, we could have selected a wild-type mice by mistake. Also, the stain could have been done incorrectly due to interference of proteins with inadequate blocking. Also, not enough hormones to test with, having experiments done on different days. Now here are some related research pertaining to my project. If you want to stop the video, you can do so and look up these related research sites, because I will not go into further detail on them. I would like to thank Nizing, my mentor, Bang and Styles, my lab coordinator, Vivian, and Jennifer, who were colleagues, Fred Wiseman, who was the coordinator of the building, Mr. Hyke, my advisor, family and friends, God, and a special thanks to Mary Zhang. Now I pulled an inspirational quote from the internet, and